All right, let's get going. Picking up where we left off last time with a discussion of the various natural forces that can create evolutionary change. And we'll pick that up with drift and gene flow and non-random mating and look at a couple of examples there. I'll also continue with a couple examples of Hardy-Weinberg calculations to help you with your lab in particular. You started your lab this week. Some of you did on the evolution section, and you saw how full they are and how busy they are. So try to use your time efficiently in those labs in order to get everything finished. And then we'll talk about natural selection specifically, the one major force that can create adaptive change in organisms. These other forces, quote-unquote forces, these are not forces like physical forces. I'm using that term somewhat metaphorically. But these other forces like drift and gene flow, they create changes in allele frequencies in populations over time, but they don't necessarily lead to adaptive change. Natural selection does, and that'll be the focus for the rest of the lecture. Hopefully we'll get to this final point today, if not next time. I'm trying to build in a bit more natural history and some videos to liven things up a bit. I lost a few of you last time, I noticed here in the classroom, so we'll have a better time with it today. I showed this slide, I believe, and defined microevolution, but I didn't really discuss all the old white gentlemen here on the slide who are considered the architects of neo-Darwinism, or the modern synthesis, which was this grand unification that synthesized Mendelian insights with Darwinian biology and mathematical modeling into a coherent framework for studying evolutionary biology. And it came to be known as the modern synthesis. Julian Huxley coined that phrase, and it stuck. People like Ernst Mayr and Theodosius Dobzhansky and Sewell Wright and Fisher and Haldane, these towering figures in the history of biology, created the framework for biology for the rest of the 20th century in this modern synthesis. In many ways, what they outlined was the study of microevolution, the study of changes at population level, changes within species, changes above the species level. The big changes in the evolution of life on this planet, sometimes called macroevolution, was largely left out of this synthesis. And that was a criticism that was leveled against the synthesis in the 1960s, 1970s, and later. And so hopefully in this section I'll be giving you a more balanced view with plenty of information on macroevolution as well as microevolution. I mentioned mutation briefly. Remember, I drew a little schematic and showed one allele changing in the population to simply illustrate the way mutation can change allele frequencies in a single generation. Now, mutation is rare. It's relatively rare, and so it doesn't change allele frequencies to a significant degree quickly. But it adds up over time, and indeed, mutation is the only way for truly new alleles to enter a population. It's the ultimate source of novelty at the genetic level. I'll go through these other factors, forces now. Genetic drift, discussed this in office hours a little bit, and described it as a random process. Random relative to the organisms themselves, or random relative to the genetic composition of the organisms. For a simple example here of using our friendly white, red, and pink flowers, you can imagine a starting population of such flowers with their allelic contributions illustrated according to the terminology that we used last time, CRCR and CRCW and so forth. So imagine these flowers in this generation, And the ones in the white boxes are the ones that reproduce successfully in that generation. It just so happens that these others don't reproduce successfully. Maybe a herbivore came through and ate the reproductive parts on those, and not these, just because the herbivore walked this way and didn't bother to step over and eat those along its way. It ate that, and only these were the breeders. This led to the next generation and this population with this configuration of genotypes. And in this generation, only these two plants contributed to the next generation. The herbivore this time walked through here and munched and indeed destroyed all of these plants. Just by chance. It just didn't happen to, it was walking through here, it's a deer, it looks over its left shoulder because there's a coyote that barks over here, and it doesn't see this plant. It's just chance relative to the flowers themselves as to which ones got eaten by the deer. It's not necessarily chancy what the deer did. There's the dog over here that barks. There's the lay of the ground. So I'm not saying it's random ultimately. It's random with respect to the flowers. Well, what does that create if only these two breed? It creates a population of all red flowers because this one was homozygous for the CR allele. And if you only have the two CR plants breeding, you're going to fix the allele for CR at that locus in the next generation completely. And if this is your entire species, which is not likely to be, this is just a population, but say it is your whole species, you've lost that allelic diversity at that locus. CW is no longer present in this population or in this species. And to get it back, well, presumably you would need another mutation event to retrieve it. But if it is just a population within a larger set of populations, within a meta-population, well, gene flow, the arrival of another seed from another area might bring the CW allele back into this local population, for example. Genetic drift. Drift you'll get to look at closely in lab. You have a really powerful way of looking at that using a computer simulation that was written by a professor in the department. And you'll be able to look at the effect of population size on drift. Population size is usually denoted just by the letter N, little case n or big N, capital N. Your starting population size is important. In the example from flowers there, that was a small population. What was it, 10 individuals or something? Well, when the population is small, chance factors like a grazing deer can influence the population quickly. In a very large population of flowers, for example, those chance factors don't have as great an effect as quickly. And so what these simulations show is that point, and you'll be able to see this in lab. This is a starting population with 20 individuals only. And chance factors, when you run a random model, shows how alleles can drift to fixation, either with 100% occurrence or 0% occurrence, just by randomly drifting over time. Sometimes it's called the drunkard's walk, right? This wobbling of passage as the drunkard walks down the middle of the street. Are they going to end up at the buildings on this side or at the buildings on that side? Well, it's a theoretically random process. Mathematicians sometimes call it that. Here's a larger population starting with 200 individuals and an even larger one starting with 2,000 individuals. In these cases, the random walk here does not lead to fixation of the alleles over the number of generations that you run your simulation. If you ran this thing out far enough, if you ran it out not for 50 generations as it's done here, but 50 million generations, you might have one of these lines randomly walk to fixation. But it's much more likely to happen in a small population. So hopefully that'll get well illustrated for you in lab. Within genetic drift, there's a couple of different phenomena that can be categorized under drift. For example, the founder effect, which is interesting to think about because it happens a lot, relatively speaking. And one of the most well-known textbook cases is among the Amish of Pennsylvania, Ohio, who were fleeing religious persecution in Switzerland before coming to America. And in coming to America, they came as a relatively small group of people with a very tight-knit community, such that they weren't marrying and breeding outside of their community to too great an extent. And those founding members of the population, there was an allele that can create a health syndrome called Ellis von Crebold syndrome, that when the individual is homozygous recessive for this syndrome, there are various phenotypic effects associated with it. Not fatal, but visible. Because the founding members of this small population were high in the number of carriers of this allele, just because of that narrowing of the population as it left Europe and arrived in the States with by chance, high number of individuals with this allele, the population as it grew over time ended up being very high in carrying this allele relative to the global human population. So you have a high incidence of this disease in the population as a result of this founder effect, where a small number of individuals founds a new population with a random array of genes relative to the original population. It's very much what is called the bottleneck effect in biology. And this is the notion of a large population being narrowed to a very small population before recovering to a somewhat larger size. The metaphor is of a bottle's neck, the population being driven through the neck of the bottle and narrowing before becoming larger on the other side later in time. And if you imagine a jar full of differently colored balls, well, what balls get shaken out through the narrow, what do you call that? Stem, is that what it is? Of the bottle? Neck of the bottle. Yeah, it's the neck. It's the bottleneck. Duh. Which balls fall out when you randomly shake it? Well, in this case, no yellows, just a few blues and a white. Because in that small number of individuals, just by chance, just by rolling the dice there, you had only a selection of those original alleles. The balls here representing the gene pool, the alleles in the gene pool. So this can be categorized under genetic drift. And there are some key real-world examples of bottlenecking, especially in light of human disruption of habitats on the Earth. Many populations have gone through bottlenecks as a result of being highly reduced in size. Not going extinct completely, but being highly reduced in size and numbers. Reducing allelic variation. And the population's experiencing the consequences of reduced genetic diversity. Here's an example from the greater prairie chicken. This humble bird of the Midwest is actually a relatively famous bird. It just became more famous recently in light of decisions around the Keystone Pipeline, because the Keystone Pipeline was proposed to be built through areas where one of these prairie chickens lived. I believe it was the lesser prairie chicken, not the greater, but I may be mistaken. But a gentleman from Kansas proposed delisting the prairie chicken from the endangered species list to facilitate the building of and the modifications of habitats in the building of the pipeline. But that was rejected in the Senate, the notion to delist the prairie chicken. This is a bird that has suffered at the hands of habitat loss quite dramatically. Illinois, shown here, the original range of the greater prairie chicken in green in Illinois, reduced to just a couple of populations because these prairies are highly desirable as agricultural fields. They're fertile areas for human agriculture, and they're converted, and the prairie chicken's habitat is lost, and all the birds die or can't find a place to live. That's the overarching point here. But within the context of the bottleneck effect, imagine the loss of diversity, genetic diversity in this population as a whole, when you go down to just these couple populations. It's measurable. The number of alleles per locus in Illinois was measured at 5.2 alleles per locus on average in Illinois before the 1960s. By 1993, the number of alleles per locus was down to 3.7. Well, what does that mean for the biology of the creature? In this case, it means a loss of fertility, a loss of reproductive success, because the eggs hatched per individual were seen to decline relatively steeply in those populations, where they didn't decline in neighboring states where the populations were not reduced as dramatically. So something about the loss of allelic diversity led to the difficulty of these birds to reproduce successfully, although that wasn't identified in the study. The correlation is there, and that would require additional effort to determine exactly what effect the genes are having on reproduction in terms of the mechanics of the relationship. You guys hear me okay in the back today? A local example, and this is a creature I'll come back to probably later in the lecture. It's a creature that I find very interesting, one called the elephant seal. You have elephant seals available to you for your viewing pleasure at Point Reyes, just on the coast north of San Francisco, Año Nuevo, north of Santa Cruz, south of Half Moon Bay. These are incredibly massive pinnipeds. They're seals with this long nose that gives them their name, elephant seal. An animal, a species that was reduced only to a couple dozen individuals in the world. It was thought to be extinct, but then if these couple dozen individuals were seen on an island off of Mexico, and the Mexican government established a conservation of them, and from those founding individuals, the population has recovered spectacularly with an exponential growth since that time, and now these beaches that I described in Northern California are some of the largest populations in the world for the species as it continues its recovery. But having narrowed to just a couple dozen individuals, you can just think about the loss of diversity and the potential consequences of that on the population. And that's being investigated now. Just give it to you as a quick example of another local bottleneck. Jumping to gene flow, a famous case of gene flow whereby simply the movement of individuals or the reproductive parts, the seed or the pollen from the plants, between one population and another can change allelic diversity in a local population. It's pretty simple to understand. If you have a local population without a certain allele, but the plants over on the neighboring ridge have that allele, and the pollen from one of those plants is carried over on a bee or by the wind into that local population with that novel allele relative to the local population, allelic diversity is going to change locally. It's going to be microevolution by our definition. In this example, you have a type of grass living on abandoned copper mines that evolved locally to tolerate copper. The alleles are identifiable, the ones that allow it to tolerate that heavy metal, high concentrations of that heavy metal in the soil. Those alleles occur in relative abundance on the old abandoned minefields. I should use my pointer here so people at home can see what I'm pointing to. So here on the mine site, you can see this high number of alleles relative to copper tolerance. Well, downwind, relative to the prevailing wind, those alleles are also escaping the mine itself and getting themselves into the populations downwind. So these individuals are taking on this copper tolerance, even though the soils here are not high in copper. All the high copper concentrations are here. So just by gene flow, just by the movement of alleles on the wind within the pollen, you get microevolutionary changes in these subpopulations. And one of our final one to look at before we get to natural selection, non-random mating. Remember Hardy-Weinberg, one of the assumptions of Hardy-Weinberg was random mating in the population. Well, non-random mating happens frequently in organisms. Organisms that breed like, with similar phenotypes, breeding together more preferentially than organisms with differing phenotypes. In humans, we know cross-culturally that there are problems with breeding like with like, specifically breeding within families because of the negative consequences genetically of breeding with very close relatives. Because you're losing allelic diversity, you're creating a greater percentage of homozygous recessives. Recessive deleterious alleles, alleles with negative consequences, are more likely to come together when individuals are very closely related. Inbreeding depression or something like this. So inbreeding reduces fitness in humans. A table to illustrate that based on first cousin marriages and showing the higher mortality rates in the children of first cousins relative to children of non-relatives. And that's just first cousins. Remember, Darwin married his first cousin. It used to be much more common than it is today. But cross-culturally, marriages between siblings, for example, are usually not approved of. And it's based, in part, in this biology because of the potentially severe consequences for health of the offspring of siblings, for example, or the closely related. An opportunity to look at one more Hardy-Weinberg equation. I want to give you at least a couple examples here on the board for you to play with. And I just drew one up relative to a plant. Let's see if I can turn on the light there. Thank you very much. A plant, a hypothetical plant that selfs. That term must have come up in your last section, right? A plant that breeds with itself. Now, you can't get more inbred than that, right? Than breeding with yourself. But a lot of plants do that, as you learned last section. So let me just give you some numbers here to play with, and I'll do this again, I think, a bit later in this lecture, so that you can practice a little bit with Hardy-Weinberg equations. I'll also give you practice questions on this and other aspects of the evolution lectures, probably by this weekend. But one of the starting points you're often given in these types of problem are the frequencies of different genotypes in a population. Now stay with me here. This is where I started to lose you guys when I started to scribble on the board, boring math, last time. Try to stay with me for a little while here. And let's just use our familiar notation of capital A and lowercase a. So homozygous for the big A, homozygous for the small a, and your heterozygous individuals. The two A's representing the two different alleles in this biallelic species, or biallelic at this locus, right? So if you're given observed genotype frequencies in this population of 0.598 and 0.052 and 0.350. And the question to you is to determine whether this population is in Hardy-Weinberg equilibrium or whether it deviates from the expectations, the predictions of Hardy-Weinberg. Would you know how to begin? Many of you are yawning and falling over already. So I'll try to keep this brief, and I'll let you mostly work it out on your own time. But I worked it out. I don't need you to check your calculator necessarily, but if you want to follow along a little bit, you can. But one of the first things you should do in these circumstances is to determine your frequency of each allele in the overall population, right? And the frequency of the A allele in the population, remember we agreed to denote it by P, the variable P. And to determine that quickly from genotype frequencies like this, because there are only half of the big A alleles in your heterozygotes, you can divide this by 2 and add it to this. And you will get an estimate of your allelic frequency there. And I did that. If you want to check me, divide by 2 and and then the frequency of the little a allele, which we agreed to call Q. Now, these better sum to 1. Because remember, P plus Q has to equal 1 based on our rules of addition there. So how do you calculate Q easily and quickly from P alone? We'll just take 1 minus Q, because P is going to equal 1 minus Q, right? Just solving for P there. P is equal to 1 minus Q, and Q is equal to 1 minus P. So if you're given one of these, just solve for the other super quickly by subtracting from 1. So what are our Hardy-Weinberg expected frequencies for the genotypes, given a knowledge of your allelic frequencies? Well, we need our Hardy-Weinberg equation. We need to just recall that and the meaning of it in order to make that assessment. So our expected Hardy-Weinberg frequencies for those genotypes, well, it's going to be P squared for your AA genotype, 2PQ for your heterozygotes, and Q squared for your little a. We have P and Q, so you just have to plug them in. P squared, 0.624 times 0.624, 0.389. 2PQ, so this is P squared, 2PQ, and Q squared. 2PQ should be 0.469. And remember, these have to sum to 1. So you can subtract this from 1 or go ahead and calculate Q squared, 0.141. Those are your expected or predicted frequencies if the population is in Hardy-Weinberg equilibrium. Well, is it in Hardy-Weinberg equilibrium? Just eyeballing it, you can compare your actual genotype frequencies to your predicted frequencies. Well, predicted, you should have almost half the population as heterozygotes, 0.46, 46%, 47% as heterozygotes, when in fact, you only have 5%. So clearly it's highly skewed from Hardy-Weinberg conditions, your actual population. And we'll only take them to show evidence for Hardy-Weinberg equilibrium if they're exactly the same. Statistically, these can be shown to be different, and they're very different by eyeballing it. Even 39% versus 59 or 60% is greatly different. Well, what's happening here? Why so little diversity of heterozygotes? Why so little why so few numbers of heterozygotes? Well, it's a result of this selfing. It's a result of non-random mating. You're losing allelic diversity. You're losing genotype diversity in having such a few number of these heterozygotes. Happens a lot in populations that don't mate randomly. That type of the commonness of the homozygous forms, which is deleterious often if your alleles are recessive and expressed only in the homozygous condition that are deleterious. All right. Maybe one more example, but it might be the end of you if I do that again on the board here. Natural selection now, the hallmark of really the Darwinian and Wallacean theory. Darwin and Wallace weren't talking about drift or gene flow, although maybe they moved around those concepts to some degree. They didn't use those phrases and didn't emphasize those in terms of their significance in evolution. They were focused on natural selection as the mechanism of evolutionary change. And we recognize natural selection today as a major mechanism of evolutionary change and the mechanism that produces adaptive change. The process, evolution by means of natural selection, or evolution, that's what how they would have phrased it, or common descent, is based on the process of individuals in the population first varying in their traits, a point I made earlier, that there's a quote-unquote struggle for existence among these individuals, a point I did not stress earlier, but a point that goes back to Linnaeus and earlier, that organisms in the world are struggling with each other, either for survival or for reproductive success. And as a result of this struggle, the variants that are beneficial will differentially survive and reproduce with success. And that the favored traits, if they're heritable, will be preserved in the population. So that's the aspects of the logic of the natural selection theory, those components. And what this has a bearing on is organismal fitness, the contribution of organisms to the gene pool of following generations relative to other organisms of their species. If an organism has 10 offspring that are successful, 10 babies that are successful, well, is that fit or not fit? Is that high fitness or not? 10. There's no scale on which to measure that. You need to look at other organisms in the population. If everyone else in the population is having 100 successful offspring, 10 is not very successful. So fitness is always a relative measure relative to the other organisms. Adaptation is, as we described, the fact of organisms having structures, having phenotypes that make them match their environments in ways that make them successful and effective. Populations vary in their phenotypic traits as a rule. And often that phenotypic variation occurs according to a normal distribution of phenotypic traits. If your phenotypic trait is on the x-axis here with a continuous variable, and the number of individuals in this histogram are shown on the y-axis here, well, the most individuals occur at some middle value in this population, leading to a bell-shaped curve. Or if we took all of us outside and lined up according to the heights of individuals, it'd be awfully fun to do. We'd probably end up with a distribution, normally distributed population like this in terms of height, with some small and some very tall, but most average in height. The bell curve, or normal distribution of phenotypic variation. To give you an illustration of how selection can influence the normal distribution of phenotypic variation, we can look at mice, which you're becoming familiar with in lab, white mice and dark mice on a spectrum with brownish mice in between. Well, if your starting population is distributed in this way, with few whites and few darks and mostly intermediates, and then some force of natural selection acts on the population against... here's your force, sorry. I was confused because I was thinking of that arrow as being the force. No, this white arrow illustrates the force. The force is acting against individuals at this part of the curve. That leads to a moving of the normal distribution to the right, leading to a change in the mean value of the phenotype to the right, toward the darker individuals. And you might imagine that a volcano explodes and the substrate becomes darker as a result of lava exposures, exposing these animals to higher predation rates, which is the example you'll play with in lab, real-world example from the southwest U.S. Or maybe your force is acting on the median, the mean values here in the population, leading to a bimodal distribution of traits in your subsequent generation with a larger number of light-colored individuals and a larger number of dark individuals, but fewer intermediates. Disruptive selection as opposed to directional selection. And this directional selection, as you might imagine, could act on this end of the spectrum or this end, pushing it one way or the other. Both would be directional. or a type of stabilizing selection that acts on the extremes, not changing the mean value of the phenotype, but reducing the variation around the mean as more individuals survive or successfully reproduce at the mean. Real-world examples of these cases. Directional selection, these swallows were surveyed during and after a very cold winter when many of them died. The investigators simply picked up dead birds off the ground that hadn't survived the cold and measured them and developed an index for their size based on their wing size and their beak size, an overall measure of the size of these birds according to these body size classes. And they looked at the ones that didn't survive versus the one that did survive. The ones that did survive the cold snap were larger. The big birds survived the cold snap, and the subsequent population was thus skewed by directional selection toward larger body sizes. I'll let you think about why big birds might have survived the cold snap. You can think of physiological reasons, maybe. Stabilizing selection from our own midst here with human infants, human body weight, birth weight, somewhere around 7 pounds as an average birth weight in pounds here. Well, the mortality curve of humans that are born very, very small or very, very large looks like this. You have a higher chance of mortality if born extremely small or extremely large. And that serves to help stabilize offspring at intermediate birth weights as a result of stabilizing selection. It's not to say individuals born here and here can't become healthy individuals. I had a Bio 1B student who, she told me I could talk about her. She was born at like 3 pounds or something, super small. Her mom went into labor prematurely up in the Sierra Nevada when they were camping in the Sierra Nevadas and had to rush out. She was born as this tiny little thing. This is a little peanut. And she went on to be on the fencing team here at Cal, and totally healthy adult as this tiny thing. But it's true that the mortality curve does look like that and helps to stabilize human populations in terms of birth weight. Disruptive selection, the one that leads to bimodality, is in evidence in a great case in West Africa from this bird, the seed cracker, which during the dry season, when resources are hard to find, birds with relatively small beaks are favored, as are birds with relatively large beaks, because they're able to exploit a different set of resources more successfully than the birds of intermediate beak size. And that can lead to generations subsequently that have larger and smaller beaks, respectively, rather than the intermediate type. So a real-world example of disruptive selection in seed crackers. Let's watch some TV to wake ourselves up here. Let me see if I can get audio.
This fish is a predator. Its bold black and white banding mimics the coloration of the species upon which it preys. Still another kind of cichlid. It is a classic con artist, dressing like its victims to get in close before ripping them off. Luckily for the prey, it eats only their scales. It's a sophisticated predator, for its prey is not killed, but merely grazed.
All right. What just happened there? So this fish, it's a cichlid. It's one of these little fishes that occurs in such great diversity in African freshwater bodies. These are also popular in the pet trade, cichlids in the pet shops. Many of the colorful fish in the freshwater aquaria will be cichlids. Well, this one looks like the other one with black stripes that had a little bit more of a gray tinge to it. This one eats the scales off of the other one. So it resembles it so that the other species mistakes this one for being one of its own kind, but that allows it to get close, and then they nail it on the left or right side and bite off a scale. So they just graze on the body of the other fish. They're not eating the other fish, they're just biting scales off, and scales have a bunch of organic material associated with them that they can live on. And if you look at the gut of this thing, it'll just be lined with scale after scale of its host fish, right? So the implications of that were studied in one of these African lakes under somewhat difficult conditions. Now, these fish that are the biters, they're either right-mouthed or left-mouthed, with the mouth literally being curved toward the host fish, depending on whether they hit them from the left side or the right side. And that's genetically based. Whether you're a left-mouthed fish or a right-mouthed fish in this population depends on one genetic locus. And there are some actual left and right-mouthed fish if you want to trigger your imagination on what they look like. Well, what these investigators did was to follow across the years, and it's a very inadequate sampling, very inadequate set of data, but it was collected under difficult conditions and has received some subsequent support. But basically, in any particular year when the left-mouthed individuals are more frequent, it's followed by years where they're less frequent. And then they're more frequent again, and then less frequent again. So they wobble back and forth, taking the left-mouthed individuals. It would apply to the right-mouthed individuals too. So in years when the left-mouthed individuals are common, they seem to not do as well in the subsequent season. So there's a maintenance of diversity in this population at this genetic locus, and a balance in left and right-mouthed individuals. Now, why might left-mouthed individuals, when they become too numerous, have less success? Can you give me one or two explanations? Sure. Yeah, so they might, exactly, one good reason. They might limit the resources available to them if they're all attacking from the left. Well, the scales on the left sides of these poor fish are just depleted. There's not enough food for left-mouthed individuals, and they might suffer the consequences in the next generation. How about another reason? Something else come to mind? The prey, I mean, if these fish are sophisticated enough, the prey could start to realize, I'm being nailed from the left side, and I need to start watching my left. And when they do that, they open themselves up on the right. And the right-mouthed individuals become more successful because their hosts are less protected on that side. Yeah, both factors may be at play, and maybe other factors that you could envision. Another example from Africa, in a really difficult disease that humans experience called sickle cell anemia, which you've no doubt heard of. Let me make a couple points about that and then give you a few numbers to play with for looking at Hardy-Weinberg relationships in a population in West Africa. Sickle cell anemia, the details are not critical here, but we can recognize that individuals in the darker reds here are more likely to be carriers of the sickle cell allele. So the sickle cell alleles are present at higher concentrations in the darker shaded areas, less common in the areas shaded in light colors. Individuals that are homozygous recessive for the sickle cell allele have very strong health consequences as a result, and it can be fatal. Individuals that have the quote-unquote normal allele at this locus have normal hemoglobin, because this influences your blood cells, and there's no consequence. individuals who are heterozygous for this allele have some effects on the hemoglobin. The shape of the red blood cells changes to a more sickling shape, but not in the great numbers that occurs in the homozygous recessive sickle cell individuals. The heterozygous condition appears to confer an advantage to individuals in these areas. And the connection is with malaria, because individuals who are heterozygous for sickle cell anemia actually are less susceptible to malaria. The symptoms of malaria are not as powerful. So there's a selection in favor of the sickle cell allele in these areas, even though it has such negative consequences in homozygous recessive individuals. And investigators calculated, and I'll give you some numbers and ask you to play with them. We can discuss them in office hours, or if something doesn't work for you, we can do it on email. But let me just show you what these authors, Cavalli, Sforza, and Bodmer, determined for populations of adults. So adult human populations, adult human pops in West Africa for, we'll call AA, AS, and SS. And SS being your homozygous recessive condition for sickle cell disease, AS being the heterozygous condition, AA being the normal hemoglobin configuration. And you'll see up on the top that it's a single point mutation that leads to the mutant hemoglobin DNA. Just that single change from a T to an A, leading downstream to a difference in glutamate and valine in the hemoglobin that accounts for the difference. Just ultimately a single point mutation at one nucleotide leading to this change. Well, what they found in this population was that 25,374 individuals were homozygous for the normal hemoglobin allele there. 5,482 heterozygotes. So these are genotype frequencies. They're genotype counts. Sorry, they're not frequencies. They're just the raw counts, numbers of individuals. Genotype counts observed, measured in the field. And then 67 individuals that had the homozygous recessive condition. These are adults, remember. And I said that the disease can be fatal. So you can just from these numbers, from these observed genotype counts, calculate ultimately your Hardy-Weinberg expected counts of individuals. You need to go through a calculation maybe of allelic frequencies. So you'll need to convert from counts to frequencies in the process. And ultimately compare your expected counts of individuals to your observed counts and determine whether the population here is in Hardy-Weinberg equilibrium or not. And remember, for our case, it basically has to match exactly. If it's varying to any significant degree, it's not in equilibrium. And if it's not in equilibrium, it begs an explanation. Because Hardy-Weinberg tests the null model of no interesting evolutionary change occurring in the population. If your expectations, your predictions differ from the observed data, something interesting is happening in the population. So please give that a try, and we'll regroup and try to catch up on what? Friday.